Welcome. In this session, firstly, I'll discuss about competent host, in which we will see how to introduce that rDNA, that whatever we have constructed, into the host cells. Then later, I'll be discussing various steps in recombinant DNA technology, that is the process of recombinant DNA technology. So let's begin this session with the competent host. So this is for transformation with the rDNA, right? So after constructing these rDNA molecules, we need to introduce into the bacterial cells. Say this is the rDNA and it has that gene of interest. So it has the gene of interest. This is the rDNA. And once we constructed this rDNA, we need to introduce into the host cell. So this is a bacterial cell, and I need to introduce into the bacterial cell so that okay, the expression of that linked gene to this plasmid will be expressed in the bacterial cell. So if it is present outside, its expression is not possible. For that linked gene or whatever the genes that you have linked with this or present in this rDNA to get expressed, they must be present inside a host cell. Okay, now there are various methods that we are employing to make the host cell to take up the rDNA. So here again, so let's see. So I have taken in a test tube the bacterial cells, and along with that I have taken rDNAs. So these are rDNAs and these are bacteria, right? Now with different procedures I will make this bacteria to take this rDNA, but not all bacteria will be able to take this rDNA. If a bacterial cell or the host cell, so bacteria or host cell, if it is able to take that rDNA, okay, this is the gene of interest here, this is the genome of the bacteria, so it has taken already this rDNA into the cell. If it is the case, then this bacteria or any other host cell, it can be plant or animal cell, is said to be transformed. It is said to be transformed, or I can say transformant. So it is the transformed host cell. If it doesn't take, then it is a non-transformant. I told you that when I'm taking the rDNA and bacteria together, okay, and I will follow that methods, those methods I'm about to discuss, right? And what happens even after following those methods, some bacteria, say this bacteria has taken this rDNA and this has also taken, but you can see there is a bacterium that did not take. So this is called non-transformant. So these two are transformants. Already they have taken that plasmid. Okay, so the difference between transformant and non-transformant is that the transformant will take up that rDNA and gets transformed, whereas non-transformant will not be taking that rDNA. So it all depends on it. Is there is no rule that all bacteria should take the rDNA, right? Now let's look at all those methods to make the host cell to take up the rDNA. So let's see method 1. So this method 1 is suitable for bacteria. So this is suitable for bacteria. So what I'm going to do in this method is, first I'm going to treat the bacterial cells with a divalent cation like calcium. So I'm going to take the bacterial cells. So all these are bacteria, and now to this I have added a divalent cation like the calcium. So I have added the calcium. Okay, it's a divalent cation, and what it does is it will, okay, make the bacterial cell walls porous. Okay, so here so say suppose this is the wall of the bacterium, and it's going to put, you know, a very small, okay, open up these small pores on the cell wall. So these are pores on cell wall of bacteria. So inside this you will find the protoplasm, of course, with the cell membrane and all, right? Okay, so first we need to treat that bacterial cells with the divalent cation. So once this is finished, so I will be incubating these cells, okay, on ice. So later I'll put this bacteria, okay, on a ice. So here, right, so these are the bacteria which I have taken and I have put on ice. So after treating with the divalent cation, okay, so here actually the calcium will increase the permeability of these cells, that is bacterial cells, permeability to you know, these rDNAs. Okay, then after treating with the divalent cation, so what I did, so this is the first, okay, step. Then in the second step I have placed that bacteria on ice. Now we need to do, right, a third step where I'm going to transfer this bacteria, right, onto a heat block. So where this will be, the heat will be supplied at 42 degrees Celsius. So these are the bacteria, and these bacteria are subjected to briefly at 42 degrees Celsius. So this is called the heat shock. Is called heat shock. Once that heat shock is finished, I will be moving on to the fourth step. In this case, again, I'll put this bacteria on ice. Okay, so first with the divalent cation treating, then later I need to put it on ice, and then these bacteria will be shifted onto a heat plate that will be maintained at 42 degrees Celsius, and that process we call it heat shock. And immediately I'll be putting that bacteria back on ice. I'm sorry, uh, back on this. So these are bacteria. Now when I do, right, this will completely make the bacterium or all the bacteria to take up the plasmids or those rDNAs. Okay, so here later, of course, this I need to put here along with that rDNAs. So these are all rDNAs. Okay, now the bacteria, whatever they are present, they will be taking up this. So this I will show up here. Now inside the bacterial cells you will find the rDNA, but all will not be taking, we will be seeing some bacteria without that rDNA. So they are the non-transformants. So these are non-transformants and these are transformants. Okay, so transformants and non-transformants. So this is a method that is especially suitable for bacteria. Now let's see the next method. So this method is suitable for animals. So method 2 is suitable for animals. So now this method is a method called micro injection, right? So here what I'm going to do, the rDNAs are introduced into the animal cells. Okay, so here I'm going to take on a slide the animal cell. Okay, so here there is a cell holder, so that keeps the cell in position. Okay, so this is the animal cell and this is a slide, and I will be doing this process under a microscope, and I will be taking a micro needle on which or into which the rDNA is loaded, and that will be injected into, so this is a micro needle loaded with the rDNA, and that I'm going to inject directly into the animal cell. Okay, so this is what I say the micro injection, because animal cells don't have the cell wall, and it will be very easy to use this, okay, micro injection procedure to introduce the rDNA into the animal cell, right? So this is method 2, which is suitable for animal cells. Then next we'll see another method, method 3. This is suitable for plant cells, is suitable for plant cells. So what this method, it's called biolistics. So it's called biolistics, or simply called a gene gun. Biolistics or gene gun. So what I'm going to do, so there is a gene gun, so into which they will be loading, bullets that will be made up of the gold or tungsten. Okay, so they are going to load particles. Of course the bullets will be very much here, I mean that bullet, so they are tiny particles, right? So they are tiny particles that are made up of gold or tungsten materials. So made up of gold or tungsten materials, and onto this gold or tungsten they are going to, okay, attach the rDNAs. So to this gold or tungsten particles they attach rDNA, right? And that will be in fact loaded into, right, a gene gun, and that gene gun when used, so here is a plant cell, so right, so plant cells will be having cell walls, okay, and right, so micro injection will be difficult to use on the plant cell, so hence we will be using a gene gun, so that in fact loaded with you know the gold or tungsten particles that attach with the rDNA. So now when they are bombarded, so this this is a golden particle with the rDNA on it, with the rDNA on it. So once this is bombarded, so directly means the plant cell is bombarded, then it directly deliver that. Okay, so this is rDNA and this is gold particle, right? So that we can directly allow this rDNA into the plant cells, right? So this is a high velocity, at high velocity, and these are micro particles. So they are micro particles. So gold particle, this is a micro particle, very tiny particle that can enter into the cell. Okay, I hope we know that how small the cells are, we cannot see it with our naked eyes. So those gold particles need to be much more smaller so as to enter into the plant cells, right? And they will be carrying on them the rDNA and finally delivering the rDNA into the host cell. So this is a method which is suitable for plants, and it's called biolistics or the gene gun. The next one, and another method, is the method 4, of course. It is suitable for both plants and animals. So in this method, I'm going to use disarmed pathogens. I'm going to use these disarmed pathogens. So disarmed pathogens means those pathogens, like for example, already we have taken agrobacterium tumefaciens, in which there is Ti plasmid. So the Ti plasmid from agrobacterium tumefaciens. So this one from the agrobacterium tumefaciens, that is Ti plasmid. So we have disarmed, we have removed the tDNA part of this Ti plasmid and allowed this to enter the plant cells. So here these Ti plasmids are usually applied for plant cells for transforming plant cells, a Ti plasmid. So we have removed that T plasmid from, and this is now the Ti plasmid is a disarmed vector. It is a disarmed vector. Okay, and right, for animals, I told you that we will be using something called the retroviruses. Retroviruses. So these retroviruses are disarmed. We removed all the genes that can cause diseases and use these retroviruses for delivering. So, or we will be using as vectors. So used as vectors for carrying gene of interest. Right. So these are the four methods, okay, for that is what the competent host for transformation with rDNA. So how we will be passing that rDNAs, that what we have constructed, into the host cells. So that is what we have seen, four methods which I have discussed here. Okay, so that's of course there are other methods that we will discuss it later. So the next one we'll see the process of rDNA technology. Process of rDNA technology. And let's see what are all the steps of this rDNA technology. So first one is isolation of genetic material. So I told you in earlier sessions, okay, to get that gene of interest, so first we need to isolate the DNA from the target cell. Okay, so first isolation of the genetic material, then the next step will be cutting DNA at specific locations. So cutting DNA at specific locations, right? Then the third step will be amplification of gene of interest using PCR. So I'll discuss that PCR in detail. Okay. Then insertion of rDNA into the host, insertion of rDNA into the host, and then okay, obtaining foreign gene product. So obtaining the foreign gene product. So these are the various steps in rDNA technology. So now first let's see the first step, that is isolation of genetic material. How we will be isolating the genetic material, okay, from the cells. So here I told you already, uh, to obtain the genes from the particular cell. Say suppose I want an insulin gene, I want insulin gene, so I can get this insulin gene from the the cells, that is pancreatic cells. So from the pancreatic cells I need to get this insulin. So I need to take the pancreatic cell, and so this, in the pancreatic cell you may find, of course it's a eukaryotic cell, and you will find cell membrane, you will find nuclear membrane, and inside that the DNA, that is the genetic material. And this DNA will not be alone, it will be associated with the RNAs and many other proteins. We need to remove all those RNAs, we need to remove all those proteins, and we need to obtain that genetic material in its pure form, right? Fine, so I'm going to obtain this insulin gene, that's what, from the beta cells of islets of Langerhans. Just I'm giving an example for that. Okay, so of course to remove the cell membrane, to remove the nuclear membrane, and to remove the RNAs and other proteins, I will be using, okay, various enzymes. So here various enzymes are employed, various enzymes are employed, okay, to remove various substances in the cell, okay, including the cell membranes and whatnot. So for example, in case of bacterial cells, in case of bacterial cells, I use enzyme called lysozyme. I use enzyme lysozyme, okay. And in case of plant cells, I use an enzyme called cellulase. So this cellulase will remove that, okay, cellulose layer, whatever, the cell walls and all. So for in case of plant cells, to remove that cell walls first. So even bacteria will have that cell walls that can be disrupted by using the enzyme lysozyme, and in case of plant cells by cellulases. And then in case of fungal cells, we know they have the presence of chitin in their cell walls. Hence, I will be using enzyme called chitinase. Okay, right. So then to remove the RNase, to remove RNase, I will be using enzymes called RNases. So these RNases, right, called the ribonucleases. So they are going to digest RNAs. Say they digest means they will break down these RNAs and completely remove these RNAs. Okay, and then to remove the proteins, to remove proteins. So what I will be using to remove the proteins, I will be using enzyme called protease. So this protease digests proteins. It digests proteins. Okay, so once this is all completed, okay, so if it is a bacterial cell, you need to apply lysozyme, then RNases, proteases, and other enzymes to remove, okay, various other types of substances. Once that is finished, okay, we will be obtaining a purified DNA only after treating with the chilled ethanol. So once that the DNA is treated with the, okay, chilled ethanol, okay, so the DNA gets precipitated. So you can see here, this is the DNA, and collecting that DNA from the chilled ethanol is a process called spooling, right? So collecting, okay, this precipitated DNA is what we call spooling, right? So this is the first step in the process of recombinant DNA technology, that is isolation of the genetic material. Now the second one will be cutting off DNA at specific locations. So let's see the second step, cutting of DNA at specific locations. I hope you know how we need to cut the DNA at specific locations, and this, for this process we need to use restriction endonucleases. Okay, so this purified DNA we will be taking into a test tube. So here we have a purified DNA, and to this purified DNA I'm going to add the restriction endonuclease. Okay, only one type of restriction endonuclease, okay, and that will break or cut this DNA into number of fragments. Okay, so that fragments I need to isolate them by agarose gel electrophoresis. So once that DNAs are fragmented, once the DNAs are fragmented, okay, so I will be, so this is the plus and minus, and this is the gel electrophoresis. I think we already discussed about this gel electrophoresis. Okay, so these are wells into which these broken or fragmented DNA. So once restriction endonucleases are added, okay, DNA fragmentation occurs. So DNA fragmentation occurs, right? And those fragments will be loaded into these wells, and now they get separated basing on their okay size, right? And of course later we are going to apply ethidium bromide and then expose it to ultraviolet radiation. Then we will be seeing that separated DNA fragments as bright orange colored bands, and those bands will be cut out from the agarose gel in a process called elution, right? And from that we will be identifying the gene of interest. All those fragments whatever we have obtained will be linked with a plasmid, and those what we call them rDNAs will be put into the bacterial cells or into the host cells and allow them to multiply, so that they can also express that linked gene, and we will come to know that which fragments of the DNA are having which genes. Okay, so that's we call it a gene library. So there is a concept gene library that we will see it later, right? So here once we identify the gene of interest, okay, we can make multiple copies of that gene of interest. So either by allowing the bacteria to multiply, okay, and the plasmid which is present with that gene of interest will also get multiplied along with the bacterial cells or the host cells, thereby making multiple copies of that gene of interest. There is another process, okay, in which we can make over a night millions of the gene of interest, okay, into copies. So that is called the PCR technique, right? So I'm going to discuss that PCR technique in the next session, and the remaining part of this process of rDNA technology in the next session.